Okay, so where we left off is the reasons why the membrane has a charge difference across it, that the outside is more positive than the inside. And so these are the reasons. There's a difference in concentration of sodium and potassium across the membrane. High sodium concentration on the outside, but there's very few sodium channels. And so therefore sodium will diffuse in from high to low concentration. There's just not a lot of the protein channels for them to do so. So they do diffuse in. And then the potassium has a high concentration inside the cell, so it's going to want to diffuse out of the cell, and it does so, and there happens to be way more potassium channels than there are sodium, so you have way more sodiums leaving. And then you also have this negative ions that do not have protein channels to diffuse, so they're high concentration inside, but they can't get out. So therefore, those are two reasons. You have more positive charges leaving the K plus than positive charges entering the sodium ions, and so that creates negativity on the inside because more positives are leaving and they're not being replaced, and so it's less positive or more negative. The negative ions are stuck. That's the second reason. All right, and then the third reason is the sodium-potassium pump, where the ions, the sodium and potassium, will never reach equilibrium while you'll have equal concentration on either side as these are diffusing in through this membrane because of the sodium-potassium pump, which pumps the 3 Na pluses out for every 2 K pluses in. So it guarantees that there's always a high sodium concentration on the outside and a high potassium concentration on the inside, and the unequal pumping, 3 positives out for only 2 positives in, contribute again to that negative charge inside. And so we say that this is what's going on in a neuron at rest, and we call this negativity inside compared to the outside the resting potential. So the difference in electrical charge, that's the difference in charge across the membrane between two regions, is called a potential difference. So you have this difference in potential. In a resting neuron, so that means it's not doing anything, it's not communicating. In a resting neuron, the potential difference between the inside of the cell and the outside is called the resting potential. And the resting potential for a human neuron is negative 70 millivolts. Okay, so that's our resting potential. So what happens is this resting potential is disrupted when the neuron receives a signal. So when it receives a signal, a process called depolarization happens. So the neuron needs a stimulus, and that stimulus could be, if it's a sensory neuron, a change in temperature, touch, pressure, light. This could be a change in the external environment, such as the change in temperature, light, or pressure. It also could be a chemical called a neurotransmitter. So this is a chemical that's released by one cell and communicates with another cell. So the stimulus could be from a neurotransmitter from another neuron. So either one of those could stimulate a neuron to relay an electrical impulse, it's called. When a neuron is sufficiently stimulated, that means that you have to get enough stimulation. So sometimes, your neurons won't fire with just a little bit, but it has to have enough stimulation. When it's sufficiently stimulated, the resting state becomes disruptive, and the membrane becomes what we call depolarized. Notice here it goes up from negative 70 millivolts up to 30 millivolts. So the cell membrane suddenly and very briefly becomes more permeable to sodium ions. So what happens is if you think about sodium ions are in high concentration outside the cell, right? And so if the cell membrane is going to become more permeable, where are those sodium ions going to want to diffuse? They're going to want to diffuse into the cell from high to low concentration. So if we look at the next bullet, it says sodium ions rush into the cell through open sodium channels, causing the inside to now become positively charged. with respect, this says with respect to the outside, and that process is called depolarization. So when it allows these sodium ions to come in, the inside becomes more positively charged. And when it says through open sodium channels, and so these they open up, they're gated, we're going to see that they're gated channels. If the stimulus is strong enough, even more sodium gates open, increasing the inflow and making it become even more positive as more and more sodiums go in. Says this is an all or nothing event. So therefore, once it's the neuron is sufficiently stimulated, then these gates open and they come in, and more gates open, and so neighboring sodium gates are also stimulated to open. And what happens is these gates, their gated ion channels, open further down the neuron. And then that's what this says here. This occurs in a sequential chain reaction down the axon at what are called the nodes of Ranvier. At this point, before we go on to the next one, what I'd like to do is have you, if you have a blank sheet of paper, I'm going to draw a neuron with you to kind of explain this as we go along. It'll make a little bit more sense. So pause this and grab yourself a blank sheet of paper. All right, so what we have here, I'm going to draw a neuron. All right, bear with me. Here, this is going to be the dendrites. Kind of looks like a moose. It's a neuron. And then here's your axon. And then here's your synaptic terminal. All right, so this is a neuron, people. All right. So what we just talked about is this neuron becoming depolarized. So the depolarization occurs where the stimulus is detected. The stimulus is detected here at the dendrites. So this is the dendrites of the neuron. And so in the neuron, there are, let's get a different color. Well, I'll just use black again. There are gates that open and close. So this is a sodium gate, and it has a gate that's closed. Okay, and remember that I'll use that sodium, I'll just put it as a little dot here. Sodium is in high concentration, so there's high concentration of sodium outside the cell compared to inside the cell. So what happens, remember that the inside of the cell is, compared to the outside, more negative. It's negative 70 millivolts. So the inside is more negative compared to the outside. What happens is when this neuron, and I'll just focus on this, receives a stimulus. So remember we just said that the stimulus could be something in the environment, or it could be a neurotransmitter. Let's do an example with a neurotransmitter. So right here, this is a neurotransmitter. What happens is if that neurotransmitter binds to this gated channel like this, binds to it, what that triggers to happen is when that binds to it, the gate opens. So now your gate opens, right? Gate opens. And that allows sodium to come in. So sodium starts to come in, and that's why it's becoming depolarized, becoming more positive. So it's not negative 70 millivolts anymore. It's going to go all the way up to, as it says here, positive 30 millivolts. And so this can trigger more gates to open. I only drew one, but there are multiple sodium gates, and so those can open, and you get an influx of sodium ions from the outside. Okay? So then what happens next is these sodiums diffuse. So they diffuse. through here, and then eventually get to the axon. Now, one thing I didn't draw here that I'm going to draw is the myelin. So I'm going to draw a couple here. All right, so this is your myelin. This right here is your node right here, node of Ranvier. This right here is your myelin sheath, which remember is made from a Schwann cell. And so what happens is the sodium diffuses, and what happens is that the positive charge inside, so the inside here is becoming more positive because of the sodium's diffusing because of the stimulus. And what that does is trigger sodium gates at the first node. Remember the node, so this is a node right here, this is the first node here, this is the last one here. That's where the axon is exposed. So there are sodium gates here that are normally shut. And so at each node, there are sodium gates. So I'm going to put everything sodium in green here. And they're on both sides. And realistically, this is around here. They're all around. I'm just going to draw one on each node to make it less busy. So what happens is this positive charge reaches this first gate and causes this gate to open. So this gate opens, and what happens is, remember, you have a high concentration of sodium inside, so the sodium diffuses in here. So now the inside of the cell right here becomes positive. What happens next is that's going to diffuse over here and trigger the next gate, the next sodium gate to open. So these are called voltage-gated ion channels. So this opens, and now sodium can go in here. Why? Is because they open and close according to a change in voltage in terms of charge. So therefore, this one opened because the inside of the cell became more positive, which then causes more sodiums to go in here, and now the cell becomes positive over here. All right, and so that's what we just talked about with when I said that the sodium gates are open, are stimulated to open equals impulse propagation. That this occurs in a sequential chain reaction down the axon at the nodes of Ranvier. That's what's going on here. All right, so it opens here first. Charge difference causes this voltage-gated ion channel to open. Influx of sodium here, the positive charge here causes the next one to open, and so on and so forth. So then what happens next, let's go back to this, and we're going to come back to this picture in just a second, is repolarization. So after it depolarizes and becomes positive, then it repolarizes and becomes negative again. So this change in potential difference lasts only for a brief period of time because the cell quickly acts to reestablish the, what do we call it, the original negative 70, the resting potential. Rapid repolarization is important because if a nerve cell does not repolarize, it can no longer respond to stimulation. So if we don't put, after this becomes positive 30 inside here as the positives go in, if we don't reestablish it back to negative 70, then the next stimulus you get, this isn't going to happen, and so therefore we won't be able to communicate, and the nerves won't be able to communicate to the central nervous system like your brain and spinal cord, which then won't be able to transmit that signal to your motor neurons to cause your muscles to move, and so on. So it's really important to reestablish that negativity inside, that resting potential. So right after the sodium gates open and the inside becomes, what does it become? Positive. What happens is the potassium gates now open. So then, let's look at here. Let's draw potassium. Let's pick a different color. I'm going to do pink for potassium, kind of going, with what I did over here. Sodium was green. Potassium was pink, keeping with that same color thing here. There are also potassium gates here. So you might want to, that are shut. Let's draw these guys here. So the potassium gate's open. So let's look at what happens. So stimulus causes sodium to go into the cell. Inside the cell becomes positive. That triggers the first gate at the first node to open. Sodium gate opens, sodium comes in, inside the cell becomes positive. Now what happens next is kind of simultaneous. At the same time, this positive charge triggers the next sodium gate to open and the inside of the cell becomes positive. But that positive charge also triggers the potassium gate at the same node to open. So this opens. And if the potassium gate opens, guess what? Potassium can move through this. Well, where is potassium going to want to move? Well, remember, potassium is in higher concentration inside the cell. So potassium moves out. So this is going to help to reestablish that negative 70. It becomes positive inside, and now as positives leave, it's going to become more negative. But you can't have more positives coming in. So what happens next is that potassium gate next closes, or sorry, sodium gate closes. So this closes back up. So when this closes back up, sodium no longer comes in, and the potassium leaves, and the inside of the cell is no longer positive here. It goes back, and it'll go back to negative 70. So therefore, that's what this is showing you. Whoops, wrong. When it says right after the sodium gates open and the inside becomes positive, the potassium gates open, and then what do you think the sodium gates have to do? The sodium gates have to shut. So therefore, the potassium leaves the cells we just wrote, and the cell becomes negative again with respect to the outside. And this is called repolarization. It's going back to what it originally was. But notice that occurs at the node. And so now it goes back to negative and it goes back to normal. So then let's look at what happens over here. So the sodium now was rushing in. The sodium gates opened and rushed in as a result of the sodium coming in here. That's going to trigger the sodium gates to open here, and sodium rushes in here, and the inside of the cell becomes positive. At the same time that this is happening, that on this node, what's going to happen at this node to the potassium gates? The potassium gates will open. Potassium will leave from high to low concentration. What happens to the sodium gates over here? Sodium gates shut, and therefore, as the potassium gates are open, the inside of the cell becomes negative. Meanwhile, over here, the potassium gates will shut here, and it'll go back to negative 70. So therefore, both will be shut, and you won't have flow into or out here. So this goes back to negative. So this is open now. Potassium leaves. Eventually, that shuts, and no movement goes, and you'll get back to negative 70. And then over here, sodiums go in, and we're going to see that that's going to trigger something to happen at the end here that we haven't yet gone over. All right, so that is repolarization. So repolarization, at each node, depolarization and repolarization happens at each node. And that signal goes from node to node down the axon. And that movement of that signal and what happens here is called an action potential. So an action potential, the rapid sequence of changes involving depolarization and repolarization is called an action potential. And so this happens at the node. So the node, the sodium gates open, sodium goes in, it becomes positive, that's depolarization. That's going to trigger depolarization over here. Meanwhile, it also triggers repolarization with the closing of the sodium gates and the opening of the potassium gates, and positive charge leaves, and therefore it becomes negative again. Okay, and so then it'll go back to being normal, and that's one action potential, and then an action potential will occur here, depolarization, repolarization, and so on and so forth. So the action potential happens at each node, but moves down the axon. So there is a brief period of time called hyperpolarization where it becomes even more negative on the outside than the inside. This is by the time the potassium gates close, there has been more potassium outflow than necessary to establish original polarized potential. And what happens is the membrane becomes what we call hyperpolarized. And so that's when we look at this, when the potassium gate opens here and the sodium gate shuts, the potassium gate's a little bit too much potassium leaves. And so instead of just dropping it to -70, it goes to -80 and before that potassium gate shuts. All right, so the refractory period is that period of time when a neuron will not respond to a new stimulus. And so during this time, the original distribution of sodium potassium is reestablished. Once ions are returned to the resting potential, which is -70, the neuron is ready for a new stimulus. And so it can transmit a signal again. And so on. So therefore, the speed of an action potential can be affected by the diameter of the axon. So how fast that moves down here from node to node to node, the larger the diameter they found, the faster the speed, all right, as which it moves down the neuron. So let's look at this picture down here. This is a picture of what happens at each node. So you start out with, notice it says membrane potential and time. So what happens at each node, it starts out at -70 millivolts. So that's your resting potential. So number one, we have our resting potential. Then number two here, we start the depolarization process, all right? And so the inside of the cell is going to become more positive than the outside of the cell. So that means that 2 here is the inside of the cell is becoming positive, and eventually when it becomes positive enough, it triggers the first voltage-gated ion channel to open at this first node. So if you notice here, we get, when that opens, you get a huge increase in charge. So it goes up to about +30 millivolts. That's what we call the action potential, depolarization, and then repolarization. So what's happening at 3 is those sodium gates open. So the sodium gates start to open here at number 2, and then once the inside of the cell reaches this dotted line here, that dotted line is as you see here, it says the threshold. It's the membrane potential it must reach in order to trigger the voltage-gated ion channels to open. And so therefore, sodium, here's your stimulus. Sodium is coming in here. It's coming in and in and in. It's getting more and more positive. That's this part here, more and more positive. Eventually, it gets positive enough, triggers your sodium gate voltage-gated ion channels to open. These don't open until it becomes a certain level, that threshold. And so once those open, you get an influx of sodium, and therefore the inside the cell becomes positive. But then remember that that's going to trigger an action potential to start over here, where it's this node is going to start depolarizing. But simultaneously, this is going to repolarize this node over here. And how it does that is the potassium gates shut. and when the open, and the sodium gate's shut. When that potassium gate opens, all the positives leave, and since the sodium gate's shut, no more positives go in. So you have the positives leaving, which means that the inside of the cell becomes more and more and more and more negative. But remember, just for a brief period of time, it dips below that negative 70. It becomes hyperpolarized, about negative 80. During that time, it cannot receive another signal. That's called the refractory period. Eventually, that potassium gate shuts, and it returns to the negative 70. How it returns to the negative 70 has to do with what we've talked about previously in the notes, which was this part. So you have remember, you have also in the membrane, you have channels that allow sodium and potassium to diffuse from high to low concentration, and so they will diffuse back to where they were, as well as with the sodium potassium pump, and so that will pump them, the sodium's back out and the potassium's back in, because sodium's left and sodium's came in and potassium left, and so therefore your sodium potassium pump works to redistribute those. And so there are sodium channels that sodium will still diffuse in, and there are potassium channels, and remember there are more potassiums, where potassiums diffuse out. But remember that you have those sodium potassium pumps. The sodium potassium pump is going to pump, maybe we should write sodium here, and potassium. Oops, I'm kind of down here, sorry. So we have those sodium potassium pump that pumps 2 K pluses in and 3 Na pluses out. So the sodium potassium pump is going to expend energy to redistribute this and to therefore bring it back to the negative 70 from negative 80. And so that's how it goes back to the normal resting potential. So let's go back to our notes. So the conduction of action potential. So this action potential, we should write here, I'm going to write here that it occurs at each node. So this process here occurs at each node. So that an action potential will occur here, 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 and so on until you reach the end of your axon. So that at the site where the action potential is generated, an electrical current depolarizes the neighboring region of the axon membrane. And so, so what that means is at the next node, okay, what we've just talked about. Action potentials travel only in one direction toward in the end of the neuron. This is called the synaptic terminals, or sometimes called the synaptic knobs. And so therefore, our direction of movement is from cell body to synaptic terminal. And so that signal kind of jumps from node to node because that's where the gates open and close. And so, so what prevents it from going in the opposite direction is something about the sodium gates. The sodium gates actually have 2 gates on them. And so they are inactivated, like when it's negative 80, when it's a negative 80 millivolts, there's an inactivation gate, so they can't open. So it doesn't matter if you receive another stimulus. If it's at negative 80, it's not going to work. So that inactivation gate doesn't become, isn't like kind of turned off until it resumes to negative 70 millivolts with the help of the sodium potassium pump. So that's what this here is. Inactivated sodium channels behind the zone of depolarization prevent the action potential from traveling backwards, meaning that when sodium comes in here, these positive charges can't trigger these gates to open. So therefore, only the gates on the next node open, and that's because that inactivation gate is shut, okay? And so what it looks like here on this picture is how the signal is transmitted is if you have, so this is your axon, so this is like this part here. So this picture here is showing you sodium gates open. So this would be like at that first node here with the sodium gates open and sodium coming in. And notice it's showing you sodium gates on both sides. I only just showed it once here, but it's really on both sides. And then that inside of the cell, so sodium comes in. And so we can show here that there's a gate that's open. All right, the gate that's open, and sodium comes in at the node. Simultaneously, the next node, so this is the sodium gate at the next node, opens. All right, while that gate at the next node is open, the potassium gates at the first node is opening, and the potassium gates here open and allow potassium out, returning this part of the cell back to negative, and this part of the cell is becoming positive. Then, so that's the depolarization and repolarization of that node. And so we could draw here, just to illustrate this, that this would be like your myelin sheath, and so this is at one node and this is at the next node. All right, so we can do this, myelin sheath. All right, and then this is going to trigger the sodium gates at the next node to open. So if we look here, that's what's happening. So notice here that this node, everything's done, it's gone back to negative. After that sodium gate opened at this node, it triggers the sodium gate to open at the next node. So let's go ahead and draw those nodes again. So sodium gates here open, causes sodium gates at the next node to open. Simultaneously, the K plus gates on this node open, and K plus diffuses out, and it goes back to negative. So that's the repolarization part, while this node is being depolarized, and it'll continue moving down the axon until it gets to the end. So if we look here, the speed of an action potential increases with the axon's diameter. We already wrote that. Invertebrates, axons are insulated by the myelin sheath, which causes an action potential speed to increase. So it goes much faster as a result of the myelin sheath. Myelin sheaths are made by cells called oligodendrocytes in the central nervous system, and the cells that make it in the peripheral nervous system are the Schwann cells, and those are the ones that we've already talked about that wrap around the axon in the peripheral nervous system. This says action potentials are formed only at the nodes of Ranvier. So we've already talked about that, which are those gaps in the myelin sheath where those voltage-gated sodium and, I should put here, and potassium are found. So if we look here at this picture shows you that here is your structure of a neuron. These are your myelin sheaths. The exposed parts are your nodes of Ranvier, and this shows you that exposed node of Ranvier. So this is where your sodium and potassium gated channels are. I should say sodium and potassium voltage gated. Okay, and here's the Schwann cell wrapped around it, the layer of myelin and so on, and this acts as an insulator. And so what happens is that it is said that when, here's the cell body and the dendrites, when the signal is reached here and it jumps from node to node. And so that's what these arrows here is, that the signal jumps from node to node. So the change in charge here triggers the voltage-gated ion channels to open here, and then they open here. So they're actually showing you this here. So they're opening, and the inside the cell is becoming depolarized. It's becoming more positive, which then is going to trigger over here and that's what they're showing you here. The inside of the cell becomes positive. It's going to trigger the inside, this voltage gate to open, and then sodium will diffuse inside there, and so on. So it's the signal is thought to jump from node to node to node, and that happens a lot faster than if you had like a bunch of sodium voltage gated channels here that had to open and close, open and close, open and close. That these guys are pretty sensitive to that charge difference in the previous node, and so this will open, and that charge, that signal through electrical charge differences moves down the axon at a much greater rate than if you didn't have the myelin. So if the myelin, it would happen much slower without myelin. All right, so the next little part here, this is where I'm going to stop. The next part is just about what happens at the end, which is where most of you also left off in the nervous system tutorial from class. So we'll pick up on what happens at that last node and what triggers it to happen at the synaptic terminal in the next video.